Good afternoon, we are Group 6. Today we are talking about the neurobiological underpinning of empathy. Here are our contents today. We will first introduce what empathy is. There are two empathies. The first one is the emotional empathy, which is the capacity to share and feel the emotions of another person. For example, emotional contagion, share pain. The second one is cognitive empathy, which is the capacity to understand other person's perspective, thoughts, feelings. For example, theory of mind, perspective talking. Let's talk about some important brain regions. Inferior frontal gyrus is a region of the brain located in the frontal lobe. On the lateral surface of the prefrontal cortex, it is roughly situated behind your temple and above your eye socket. It is divided into key Brodmann areas, BA44, BA45, and BA47. Here are the functions of IFG. The first one is motor planning and speech production, especially in the left hemisphere. BA44 and 45 are part of Broca's area, which is essential for speech production and language processing. The second one is action understanding. The IFG helps us understand the goal and intention behind action performed by others. The last one is inhibition and cognitive control. It plays a role in stopping automatic responses and selecting appropriate behavior. Inferior parental lobe labile is a region of the brain located in the prefrontal lobe, specifically on the lower part of the lateral surface of the hemisphere. It lies just behind the postcentral gyrus and above the lateral sulcus. It is primarily composed of two Brodmann areas, BA40 and BA39. Here are the functions of IPL. The first one is sensory integration. It integrates sensory inputs to create cohesion perception of the body and its relation to the external environment. The second one is spatial reasoning. It helps with visually guided grasping and understanding the spatial relationship between objects. The last one is language and calculation. The left angular gyrus is particularly important for reading writing. Anterior cingulate cortex is a region located in the medial part of the brain. Right at the front, it forms the frontal part of the cingulate cortex, which is a bell-like structure that walks around the corpus callosum. Here are the functions of ACC. The first one is affective component, processing emotion, especially the unpleasantness and or distress associated with stimuli. The second one is the cognitive component, involved in conflict monitoring, error detection, and executive attention. The third one is the autonomic or physiological component, regulating heart rate, blood pressure, and other visceral responses in emotional situation. The last one is pain processing. It is a critical part of pain matrix, encoding unpleasant, distressing sensation of pain. Anterior insula is a region very deep within the lateral sulcus. It is the prominent fissure that separates the frontal and parietal lobe above the temporal lobe below. Here are the functions of AI. The first one is interoception. It is the primary function. The AI is responsible for sensing the internal states of the body. It creates the conscious awareness of the body signals, which is fundamental to the feeling of emotion. The second one is emotional awareness by disgust, anger, and happiness. Awareness by integrating body signal. The AI helps generate the subjective feeling of emotions. It is crucial for translating physical sensation into conscious emotional experience. The last one is risk and uncertainty. The AI is activated during task involving task decision making under uncertainty, and anticipation of negative outcome. Ventral medial prefrontal cortex is a region located in prefrontal cortex on the ventral and medial surface of the frontal lobe. It is situated just above the eye, behind the forehead. It encompasses several Brodmann area, including BA10, BA11, BA14, and BA25. Here are the functions of VNPFC. The first one is emotional regulation. It helps regulate emotional responses by inhibiting the amygdala. The second one is value-based decision making. It is essential for judging the value of choices, particularly in social and moral content. The last one is self-reflection and autobiographical memory. It is highly active when we think about ourselves, our personal past, and our traits. Dorsal medial prefrontal cortex is a region located on the medial surface of the frontal lobe. As a part of medial prefrontal cortex, it is situated in the dorsal portion of the MPFC, above the ventral medial prefrontal cortex. Here are the functions of the MPFC. The first one is mentalizing. It is critically involved in thinking about the mental status of others, their thoughts, belief, knowledge, and intention. The second one is social attention, directing attention to socially relevant stimuli and processing the traits and for other people. The last one is cognitive control in social context. It helps in making inference and reasoning about others' behavior. Temporal parietal junction is a functional region at the intersection of three brain lobes: the posterior part of the temporal lobe, the inferior part of the parietal lobe, the occipital lobe is nearby, providing visual input. Here are the functions of TPJ. The first one is theory of mind or mentalizing. It is the most well-known function. The TPJ is crucial for reasoning about the content of other people's mind, their belief, intention, and knowledge. The second one is self-other distinction. It helps distinguish between your own and other person's perspective. A fundamental requirement for accurate mentalizing. The last one is multisensory integration and attention. The right TPJ, in particular, is involved in reorientating attention to salient or unexpected stimuli in the environment. Medial temporal lobe is a system of closely related area located on the medial surface of the temporal lobe. This region is very deep within the brain near the temple. Here are the functions of MTL. The first one is episodic memory. It is essential for forming, consolidating, and retrieving autobiographical memories, the memories of specific events from your personal past. The second one is spatial memory. The hippocampus, in particular, is crucial for creating cognitive map of our environment. The last one is declarative memory. The MTL is key for conscious recall of facts and events. It is a table of components that participate in the cognitive and emotional empathy network. Each component is associated with distinct functions that comprise the empathetic response.
Let's go to the neutral circuit part. If you see a person fall dance, what will our brain happens? First of all, our temporal circuits will perceive and interpret the visual or auditory information into social intention and intention. For example, when we set our visual cortex data, a human body undergoes a certain anticipated event. Meanwhile, the Anterior insula will integrate the simulation and generate a retinal impact output to motivate the response. It acts as an emotional center to turn others' feeling and experience into ours. And the mirror neural system will understand by internal simulation, labeling the information, and provide the essential data for the rest of the empathy network to generate the feeling. Moreover, to elaborate and regulate our action and emotion, the anterior circulate cortex will receive the message from the AIG, the insula, and process the unpleasantness with the pain and provide the motivation significant to initiate our actions. And the dorsolateral prefrontal cortex will inhibit and regulate the activity of the amygdala and the AIC to prevent our overload of our empathy and control our attention to the situation. And the medial prefrontal cortex and the temporal parietal junction will recognize the social context so that the relationship between you and the person for that and his mental state. And the TPJ will distinguish the perspective taking and allow us to share the emotion without being overwhelming. To give a long-term pro-social behavior, the reward and motivation circuits will help by releasing the dopamine. It will create a sense of anticipation and positive reinforcement while we are helping others.
Let's talk about some important neurotransmitters. Oxytocin is a neuropeptide that acts as both hormone and a neurotransmitter. Its primary function, as highlighted in the article, is related to social bonding, attachment, and authoritative behavior. It is crucial for processes like mother-infant bonding, peer bonding, trust, and social approach behavior. Dopamine is a neurotransmitter that plays a critical role in several key brain functions. Its primary functions are reward, motivation, and reinforcement. Dopamine is essential to the brain reward system. It is released in response to rewarding stimuli, reinforcing behaviors, and driving motivation to pursue goals. The second one is executive function. It is critical for high-order cognitive process like working memory, cognitive facility, and inhibitory control. These functions are primarily managed by the prefrontal cortex, which is rich in dopamine receptor. The last one is motor control. Dopamine is essential for coordinating smooth, purposive movement. The process of dopamine signaling follows a standard sequence for neurotransmitter function. The first step is synthesis. Dopamine is created inside the neuron. It is synthesized from the amino acid tyrosine, which is converted into L-DOPA and then into dopamine. The second step is storage. The newly created dopamine is packed into vesicles for storage. The first step is release. When an electrical signal travels down the neuron, it triggers the vesicles to release dopamine into the synapse, the tiny gap between two neurons. The fourth step is receptor bonding. The dopamine molecules cross the synapse and bind to specialized proteins called dopamine receptor on the receiving neuron. Its bonding is like a key fitting to a lock, which triggers a change in the receiving neuron, either exciting or inhibiting. The final step is reuptake. After delivering its message, dopamine is released from the receptor. A protein called dopamine transporter then acts like a wet cues cleaner, sucking dopamine back into the original neuron to be recycled for future use. This cycle is of release, bonding, and reuptake is how dopamine transmits its signal across neural circuits.
Let's move on to the hormones part. Oxytocin not only acts as a neurotransmitter but also acts as a hormone when it's released through the bloodstream. It is stored in the pituitary gland. When signals, the gland will release oxytocin slowly direct into the bloodstream. It can increase our emotional empathy, and it can significantly enhance our social reinforcement learning. From a study from Hartman's, and oxytocin treatment can significantly enhance learning that is social reinforced. In addition, it can increase emotional empathy, raising in men up to the higher level normally seen in untreated women. Next is cortisol. This is a stress response system hormones. Generally, stress is reduced when under pressure. The brain will send a signal to the body through the HPA axis. This system can tell the adrenal gland to release cortisol, helping us to gear up for action. Cortisol can help us to impair our cognitive facility, weaken people's ability to engage in goal-direct self-control, which help us to give attention to the situation such as when the people fall dead, and amplify our emotional attention to ensure adequate behavior toward the related groups.
Moving on to case studies in supportive literature. Firstly, a case study on nursing to explain and define empathy. In nursing, they define two forms of empathy, mentioned in our introduction. These two forms are prevalent in the other studies and literature found for this presentation, as they are used as the universal definition for the two types of empathy. Firstly, emotional or affective empathy, which in the case of nursing involves sharing the patient's feelings. Secondly, cognitive empathy, which is the detached analytical understanding of their patient's experience. Using phenomenology, which is an approach based on the first-person perspective experience, we can define basic empathy as the immediate direct perception of another person's emotional state through their expressions. This integrates emotional and cognitive empathy. An example would be seeing the anger in a scowl or a fear in a flinch before an injection. You can understand these emotions and feelings without experiencing it directly yourself or needing to logically deduce these actions. Empathy is a brain or human function. A review by Schleim involving multiple case studies analyzes the relations between brain and empathy. The prominent brain regions mentioned were the anterior insula, anterior cingulate cortex, and the secondary somatosensory cortex. The case studies include Wicker et al. Finding that both feeling disgust and seeing another feel disgust activates the left anterior insula. Next, Singer et al., who found that both experiencing pain and seeing a loved one experience pain activates the anterior insula and anterior cingulate cortex. Lastly, Kazers et al., which found that being touched and seeing someone be touched activates the secondary somatosensory cortex. The problem that Schleim raises is the operationalization of empathy. Neuroscientists define empathy as brain function, but this reduces a complex human experience to simplified measurable brain signals. Empathy is not just a simple function. Many processes, mechanisms, circuits, and brain regions are used to create our actions and feelings of empathy in response to what we experience in social contexts. In this sense, the brain is the neural machinery of empathy. Empathy is complex and has evolved into a critical function in our daily lives, involving both brain processes and situational and environmental factors. A case study was conducted by Calabria et al. On a patient that had semantic dementia, characterized by the progressive loss of semantic knowledge, so words, objects, and concepts, etc., which is a specific clinical variant of frontotemporal lobar degeneration. The brain regions mentioned include the temporal lobes, particularly the anterior temporal lobes, which is crucial for cognitive empathy and semantic knowledge. Atrophy here would lead to deficits in perspective taking. And the amygdala and orbitofrontal cortex, which are key for processing and recognizing emotions from facial expressions, which is essential for empathy to recognize the state of others. Calabria et al. Notes on the right hemisphere's specialization, where the emotional aspects of empathy may be more reliant on the right temporal lobe. Bilateral temporal damage impairs both cognitive and affective empathy, whereas predominantly left-sided damage can spare affective empathy. The patient conducted the Interpersonal Reactivity Index, which is a measure of dispositional empathy, using 4 subscales: empathetic concern, the tendency to experience feelings of sympathy and compassion for unfortunate others; personal distress, the tendency to experience distress and discomfort in response to extreme distress in others; perspective taking, which is the tendency to spontaneously adopt a psychological point of view of others in everyday life; and lastly, fantasy, the tendency to imaginally transpose oneself into fictional situations. So they conducted the Ekman 60 Faces test as well, which is an instrument to test the recognition of basic facial emotions. The findings were that affective empathy was preserved and cognitive empathy was impaired. In the standard verbal version of Ekman's test, the patient was impaired in recognizing all basic emotions. This test involved presenting photographs of faces with 6 verbal labels. The patient is required to verbally select the label that best describes the facial expression shown. In the modified nonverbal version, the patient could only recognize happiness. This test involved 6 photographs displaying 6 possible basic emotions alongside a target photograph. The patient was required to choose a photograph that matched the emotional expression of the target photograph. They analyzed that there is a possible underlying language semantic deficit, as she had difficulty linking visual emotions to the correct linguistic concepts. The brain network for recognizing happiness was spared, yet circuits for recognizing negative emotions were damaged. This concludes that cognitive and affective empathy are supported by several neural substrates. Here's a clear chart of findings in the study. In the function of cognitive empathy using subscales of perspective taking and fantasy, this function was impaired. The associated region is the left anterior temporal lobe, which we know is atrophied in the patient. For emotional empathy using subscales empathic concern and personal distress, this function was preserved. The associated brain region is the right anterior temporal lobe, which is relatively intact compared to the damage in the patient's left anterior temporal lobe. For emotion word knowledge, this was tested through a short adapted questionnaire asking questions like, how would you feel if? And the patient scored 6 out of 6 correct responses. To be clear, this was a test to determine her understanding of emotion words. This was preserved. And this is associated with the semantic network of the brain, which is only partially intact in the patient. In facial emotion recognitions, in regards to the standard version of the Ekman test, this function was impaired. The associated region is also the left anterior temporal lobe, which as we know is atrophied in the patient. Lastly, facial emotion recognition in the modified version of Ekman's test, this was relatively preserved in the case of happiness. The associated brain regions found by the amygdala and orbitofrontal cortex, which are relatively intact in the patient. Moving on to empathy as a function of survival, Desetti et al. Describes empathy as an evolutionary adaptation. We had a fundamental need to care for vulnerable offspring, which drove our evolution of empathic sensitivity. We developed the ability to perceive and respond to distress signals in our offspring, which ensured the survival of said offspring. This meant passing genes that promoted this behavior. This has evolved to the modern age. In the pain system, it was found that the brain uses the same neural alarm system for social pain as physical pain. This was in the anterior cingulate cortex and anterior insula. In the reward system, the same dopamine and oxytocin-driven pathways for eating and sex also reinforce parental care and pro-social acts. This was found in the ventral striatum. This displays that empathy has adaptive flexibility and limits. Our capacity has evolved from just kin to non-kin. We feel empathy at more or less in different situations, more for in-group members, less for those we perceive are responsible for their plights, and more for those we believe are like us. However, the system fails in psychopaths. A functioning empathic system is critical for inhibiting aggression and maintaining social order. How do we experience empathy? Firstly, through bottom-up processing, which is driven by the mirror neuron system. As mentioned before, mirror neurons are the brain cells that respond or fire to actions we observe in others. They fire in the same way when we recreate the action ourselves, and they're also responsible for other sophisticated behavior and thought. The other key regions are the anterior cingulate cortex and anterior insula, which are considered part of the extended network, but not in the core system of the mirror neuron system. Secondly, top-down processing, which involves the regions of medial prefrontal cortex and temporoparietal junction. The behaviors and actions we would use would be imitation, mimicry, and helping in pro-social behavior. There are factors to our empathy, which is stimulus intensity. Stronger pain or intense emotional expressions trigger stronger empathic brain responses. Closeness and similarity, we empathize more strongly with loved ones and strangers. Context, empathy decreases if we believe pain is inflicted for a therapeutic purpose or if attention is distracted. Expertise, where it was found that professionals who frequently deal with others' pain, for example doctors and acupuncturists, can show reduced neural empathy, likely as an adaptive mechanism. Lastly, gender, where women generally show stronger neurobiological and behavioral empathy, possibly linked to the neuroanatomical differences in the MNS, and men's empathic responses are more strongly modulated by social factors like perceived fairness. They may even feel reward when an unfair person is punished. When these systems are disrupted, in autism it can lead to challenges in recognizing sharing emotions. In frontal lobe damage, it results in unregulated emotional mimicry or personal distress instead of empathy. Moving on to what happens when there's a lack of empathy, in a study of empathy profiles on violent and psychopathic criminals, yet again their cognitive and affective/emotional empathies were both measured, and found that there was a significant deficit in perspective taking compared to the control groups. Diaz-Galvin et al. Notes that the role of personal distress. Instead of distress in a response to another's, this takes form in a self-focused aversive reaction. This was found highest in violent criminals and positively correlated with psychopathy scores. So this essentially redefines personal distress into a specific form for violent and psychopathic criminals. The empathy profile was found to have a combination of low perspective taking, which implies impaired cognitive empathy, and high personal distress as a form of selfish emotional arousal, which leads to a high risk profile for severe violence and criminality. The regions mentioned in the study are the amygdala and fusiform gyrus. They found a hypoactivity in the amygdala, which results in emotion recognition deficits in psychopathy. This explains their lack of empathy, most particularly for recognizing distress cues of fear and sadness in others. Diaz-Galvin et al. Contrasted autism and psychopathy, where the empathy deficits in autism are linked to abnormal functioning of the fusiform face area, suggesting the ability to identify facial expressions affects empathic ability. We can conclude 3 types of empathy from our research. Firstly, positive, which is the capacity to share, understand, and resonate with positive emotions. Negative, the capacity to share, comprehend distress, pain, or adversity of others. Lastly, self, which is found in violent criminals and psychopaths, where they feel empathy in the form of a response to another's personal situation for personal benefit. Regenders et al. Conducted a comprehensive review of many studies in psychopathy, which is characterized by the interpersonal manipulation, affective deficits, and impulsive or antisocial behavior. The prevalence is around 1% in the general population, but a staggering 15 to 25% in prisons. Psychopaths have impaired affective empathy, but their cognitive empathy remains intact. They may use their cognitive empathy, the ability to understand a situation, to manipulate others. They have found that psychopaths have deficits in recognizing fear and sadness, and also general emotion recognition deficits. These can stem from amygdala dysfunction and intentional biases. Starting with the amygdala, firstly the subregion of the basolateral amygdala, which is associated with goal-driven behavior. Psychopaths may have a hyperactive BLA connectivity with frontal regions supporting their focus and self-interested behavior. Secondly, in the central amygdala, which is involved in automatic emotional reactivity and general motivation, psychopaths show reduced central amygdala connectivity, which could explain their lack of spontaneous emotional responses. They can process emotions when it aligns with their goals, but not when they're peripheral to their interests. With the prefrontal cortex, which is implicated in cognitive control, attention, and goal-directed behavior, psychopaths show increased functional connectivity between the BLA and prefrontal regions. This supports their superior attentional control and ability to ignore emotional information. Newman and Lorenz's response modulation theory proposes that individuals with primary psychopathy have difficulty processing emotionally arousing peripheral information because of an attentional deficit that prevents them from shifting focus away from their primary goal-directed task. This is linked to the prefrontal amygdala circuits. Moving on to the anterior cingulate cortex and anterior insula, which has been mentioned many times previously, involved in empathy, pain processing, and emotional awareness. Children and adolescents with psychopathic traits showed reduced fMRI responses in this area when viewing others in pain. This is considered a neurobiological marker for empathy deficiency. The underactivation of this region in psychopathy contributes to a lack of empathic arousal. The striatum, which is involved in reward processing and motivation, the basolateral amygdala influences motivation through the striatum. In psychopathy, this pathway may be overly dominant, reinforcing the instrumental self-serving behavior at the expense of empathic responses. Other regions briefly mentioned are the sensory-motor cortex, which integrates sensory and motor input, which in context to mimicry and emotional contagion acts as a precursor of empathy, so this region may also be involved. Lastly, the frontal limbic circuitry, which is found to be dysregulated in psychopathy. To conclude on empathic behavior, we know cognitive empathy is the understanding of others' emotions, affective emotional empathy is the sharing of others' emotions, empathy is our immediate and direct perception of others' states from expressions or contexts, and empathic behavior is the outcome of the integration of cognitive and affective empathy.
Moving on to the disorders. The first disorder that we would like to talk about is the autism spectrum disorder. So in this disorder, it is associated with reduced empathy, particularly in cognitive empathy, due to atypical brain activation in areas like medial prefrontal cortex and anterior cingulate cortex. And then we also have the narcissistic personality disorder. In this disorder, it involves a profound lack of empathy rooted with neurobiological hypersensitive to interpersonal rejection, with elevated oxidative stress markers in blood plasma correlating to diminished emotional attunement, as evidenced by reduced activation in empathy-related networks like the anterior cingulate cortex during the social interactions. From a biological lens, NPD's dark empathy manifests as cognitive empathy deficits. The next one is the borderline personality disorder. In this disorder, it talks about the neurobiology of BPD, features heightened affective empathy driven by hyperactivation in the insula and amygdala, leading to an intense emotional cognition and over-arousal to others' distress. And the next one is about the conduct disorder. So in this disorder is more associated with adolescents that shows a typical empathic response with fMRI evidence of immature frontal lobe function and reduced activation in empathy-related networks. This ties to inhibitory control deficits and altered neural processing of others' distress. And the last one is semantic dementia. In this, a neurodegenerative disorder affecting emotional recognition and empathy, with case studies showing impaired affective empathy due to degradation in semantic processing and related to brain areas such as the temporal lobe. This leads to reduced ability to stimulate others' emotional state. So the disorders emphasized on how empathy deficits can emerge from neurobiological disruptions such as in mirroring systems or cognitive control areas, which are often shaped by factors like gender, age, or hormones. So this.
All right, thank you. So an extra 3 minutes